Feriel Ramdhane, and I am image data scientist at the Medical Hub team of Euro-BioImaging in Turin. I'm going to present you the developing tool for verification of preclinical data. So let me first start by introducing the FAIR issues at the preclinical level. Unlike datasets which benefit from the well-established standards and numerous repositories, preclinical image datasets lack uniform standard and central repositories, which is challenging to aggregate data across the study. Two, there is no widely adopted metadata model for preclinical image datasets, leading to an inconsistent and incomplete data description. Three, the FAIR principles are not well implemented in the preclinical domain. So the difficulty in locating and assessing datasets lead to a duplicate of efforts, as a researcher may conduct similar experiment without knowledge of existing data. And the inability of reuse the dataset effectively reduce the overall efficiency of the research and the advancement of knowledge. Four, there is a lack of standardized protocol and tool of processing preclinical images. So the preclinical imaging is highly variable in terms of techniques and methodologies used, which complicate the comparison of results and affect the reliability of conclusion drawn from preclinical studies. And five, effort to address these issues are often isolated, so which with some initiative taken place at an individual centers, institutions, or regional national level that do not benefit from broader collaboration and standardization. At the Euro-BioImaging MEDAP, we aim to improve the verification of the preclinical image datasets by developing the PIDAR repository of metadata description for comprehensive metadata management, creating a structured metadata model curated with a selected ontology to ensure a detailed and consistent description of preclinical image datasets. We are also introducing XNAT-PIC, a free and open source Windows desktop application to streamline the management of preclinical image data. And lastly, we are leveraging the XNAT platform to develop the Decorized tool for image processing and quality assessment designed to enhance the standardization and the fairness. I will now take a closer look at each of these tools. So the first, PIDAR, the Preclinical Image Dataset Repository, is a public repository designed to catalog metadata information describing preclinical image dataset from any modality, imaging modality associated with the peer reviewing publication. So PIDAR will not function in isolation, but will connect with other major research infrastructure repositories such as MMI, InfraFrontier, and UI OpenScreen. And in PIDAR, the imaging metadata are organized as a collection defined by a common disease, image modality, or even research focus. The construction and the storage of our repository start from Excel metadata datasets, where we store and share all the information that would be filled by the data holders. Then we transform this information in SQL tables with SQL language, and we share all the metadata in PostgreSQL database. In the same database, we save the information coming from ontologies, and we make the query in the Jena Apache software with the SPA-RQL language. And then we transform an SQL table to the database with the PostgreSQL PHP function. Then the user can access to the web user interface created in HTML, Bootstrap, and CSS, and it's hosted by the University of Turin in Italy. The functionality of PIDAR, so first access to metadata related to preclinical image datasets, download the metadata in JSON format. There is a drop-down menu where user can search for specific metadata in specific field or domain, like cell lines, disease model, drugs, etc. and integrated query between ontology, synonym, and terms. There is schedule viewing for all the dataset metadata and statistical information on the store dataset. Here, some representative captures show the total dataset that are shared in PIDAR. Since PIDAR is a new repository, we have few datasets at the moment. We have only 13 datasets with 278 subjects and 192 metadata and 6 ontologies. So PIDAR is intended not actually to store images, but to store metadata that describe full preclinical image datasets, and it allows also the user to find their images, since they also added information about the dataset as well as the link where they store these metadata. The next tool is the XNAT-PIC Preclinical Imaging Center, which is a free and open source Windows desktop application to expand to XNAT core functionality, in which the adoption also of this tool will improve the FAIR principles for preclinical imaging image dataset and meet the need for preclinical image facilities. Here you can find the QR code to download our application. And the feature of XNAT-PIC, first, it's a standalone application available, of course, for downloading and immediately usable without requiring additional setup. User-friendly interface, designed easy to navigate and to understand. There is secure access where the credential to connect to an XNAT instance to load image dataset from the desktop client are protected by a cryptographic algorithm. Improve the speed of conversion raw MRI to DICOM by using multi-core processing. Interface for grouping annotation designed to allow a user to easily group and categorize information regarding preclinical images. DICOM converter for Bruker Paravision software. Also upload of project, subject, experiments, so a user can convert, can upload or create an entire project or single project or experiment on XNAT. And also there is additional that not DICOM file can be also uploaded to XNAT platform, like a parametric map or results in text format. So XNAT-PIC is intended for scientific and researcher working in the preclinical and biomedical imaging, with the aim, of course, to reduce the technical barriers associated with the data management. Then, last but not least, we developed standardized and open source tools for assessing several image quality metric through XNAT. For, of course, easy and fast evaluation of preclinical imaging datasets. We developed the SNR, the signal-to-noise ratio index, with the higher values indicating better image quality. The sharpness index, which refers to an image overall clarity and detail, with the higher value indicates sharpness images. And the mutual information, which calculate the mutual dependency. And then, of course, with the higher index, translating higher dependency between two consecutive temporal acquisition, like the DCI MRI images. We are also working to develop other index to access the quality of the images, such as the estimation ratio noise in the MRI, and the BRISC, the blend and referenceless image spatial quality evaluator index. This is how we implemented our tool through the XNAT platform. So we developed the different metric in Python. We packaged them in Docker containers through Docker files, and then we pull it and run it in the container inside XNAT using the developed commit JSON files. User here can explode this tool at the scan level or also at the subject, so the whole datasets level, by running the container with the selected quality index. And the output of this tool are saved as a plot PNG format or as Excel sheet file reporting the value of each scan here, as you can see in the right into the screen. Here are some results of the calculated SNR and sharpness index coming from tested preclinical and clinical actually dataset through XNAT. So after downloading the results executed in XNAT, we used the interquartile range, which is a measure of spread and variability in a data, and we applied the outlier rules to categorize the result in enough image quality or not. We tested on clinical MRI datasets 72 subjects, including brain and breast images with T1, T2, and T2 FLAIR, and 72 preclinical MRI subjects with T1, T2, and chemical exchange saturation transfer system MRI. From the plot, you can see that we successfully distinguish between low quality images and high quality images, especially for the data sets coming from T2 and CES data, where you can see that there is some scans in red, which are labeled with low quality in terms of SNR. And on the right here, some representative images shows the difference between low and high quality images of the SNR for both clinical and preclinical. So here it's true that we validate this tool only for MRI modality at the moment, but we are also working to validate other imaging modality, so that can benefit preclinical researcher at large scale. And by packaging this tool in Docker container, we are ensured that they are easy deployable and also reproducible. And to conclude, we encourage preclinical data holders to utilize our tools by sharing their data or metadata in PIDAR, storing and uploading their dataset through XNAT-PIC to XNAT instance, and check the quality of their data by using our quality assessment index. And of course, with the goal to enhance the producibility and the impact of preclinical research.